Now we'll move on to aim 2, where we'll characterize the pathobiological outcomes of primary blasts measured via electrophysiology. This data is published across 2 different papers, both within the Journal of Neurotrauma. So in order for us to quantify electrophysiology, we utilized a microelectrode array system, or MEA system. An image of one such microelectrode array is seen below to the right. Above, we can see a hippocampal slice placed on top of the 8x8 microelectrode array, where we have annotated the major anatomy of the hippocampal slice, including 3 main regions: the dentate gyrus, or DG, corneomonas 3, or CA3, and corneomonas 1, CA1. These regions are connected together by 2 major neuronal pathways, the mossy fiber pathway and the Schaefer collateral pathway. For this study, we exposed slice cultures to a range of blast levels, including levels 1, 2, and 4, as we used previously, and we added an additional group, level 9. These are compared to sham-exposed controls, which were treated exactly the same as the blasted groups. However, they did not receive a blast exposure. We first observed that blasts significantly reduced long-term potentiation, or LTP, which is a measure of long-term synaptic plasticity, considered by neurobiologists as the cellular correlate for learning and memory. A quick background: LTP is induced by high-frequency stimulus, or HFS, typically delivered around 100 Hz across the Schaefer collateral pathway, traditionally measured in the CA1 region. Above, we see 2 voltage signals from the same electrode when delivered the same electrical current stimulus. However, the left image is from a pre-induction LTP, and the right is from post-LTP induction. When we look at the peak-to-peak magnitudes, which are denoted by red and green in each respective image, we can see that post-induction, the peak-to-peak magnitude is clearly higher than the pre-induction magnitude. This increase is normalized to the pre-induction magnitude and then reported as the percent potentiation above baseline. So in this graph below, we see percent potentiation on the y-axis and increasing blast intensity on the x-axis. After a level 2 blast or higher, we see a significant decrease in LTP as compared to the sham group. We can contextualize our LTP data with the strain and strain rate data from AIM-1 to determine strain-based injury thresholds. We find that deficits in potentiation positively correlated with both strain and strain rate. We see a strain threshold for significant LTP deficits is between 3.7 and 6.7%. A previous study examined stretch-induced LTP deficits and did not observe changes after a single 5% 12 reciprocal second stretch, but found repeated stretches at the same intensity were able to reduce LTP. We observed a strain rate threshold between 25 and 33 reciprocal seconds. Stretch injury studies have observed that electrophysiological dysfunction exhibits complex trends with both strain and strain rate, where at low strain magnitudes like those we observe in this study, functional response can change with increasing strain rate. The strain rate dependence has also been observed with stretch-induced changes in intracellular calcium ion concentrations as well.
Quick question. So
yep.
each one of those points is a different culture?
So different blast exposure groups. So we have our shams here. This is level 1, level 2, and level 4. So you see as strains increasing with increasing blast intensity as we were using before, similar with strain rate.
And is that averaged across cultures?
Yeah, averaged across 3 cultures for the strain and rate. Since LTP was the most sensitive measure we observed, we then looked at the time course of blast-induced LTP changes following level 4 and level 9 blast, denoted in gray and white respectively. We observed that blast-induced delayed deficits in LTP. Here we're looking at percent potentiation on the y-axis and the measurement day post-exposure on the x. At day 0, neither blast intensity reduced LTP. However, at day 0, just for your information, that's recording at 1 hour post-injury. However, at 24 hours post-injury, both blast intensities significantly reduced LTP, and this deficit persisted all the way out through 6 days. However, by 10 days post-injury, we observed that level 4 exposed cultures spontaneously recovered. However, level 9 cultures still remained significantly depressed. So, to summarize, we found that primary blasts altered LTP, exhibiting a threshold for deficits between level 1 and level 2 blast, or if we consider that in terms of strain, that threshold exists between 3.7 and 6.7%, and rate threshold exists between 25 and 33 reciprocal seconds. We also investigated basal neuronal function, which we didn't show here, but we did that via spontaneous event properties, stimulus response, and paired pulse ratios. Here we found that these measures were minimally altered by blast exposure, and that LTP was the most sensitive measure to primary blast. Any functional change we observed occurred with minimal concomitant cell death, which has been supported in both blast and non-blast TBI literature. From the time course of LTP deficits following blast, we observed that deficits were delayed, developing between 1 and 24 hours. However, there was potential for spontaneous recovery between 6 and 10 days, depending on the exposure level. Now, while some physiological meaning can be extracted from these measures, it's difficult to extrapolate electrophysiological dysfunction up to changes in whole animal behavior. Thus, translational studies will be necessary to determine how primary blast influences behavior, and specifically hippocampal-dependent behavior.
I can go back one
Yep.
slide. So in here, one question. So each day you use a fresh slices, or you use
So
one
it's-
slice?
No, the fresh, different slices.
Different
So,
slices.
yep. So, yeah, because when we do our electrophysiology process, we have to excise them from the membrane, and we haven't tried to keep them alive after they've been placed onto the microelectrode arrays. Mhm. So now we'll move on to aim 3, where we will explore the effect of primary blast exposure on LTP induction pathways. Again, this work is published in the Journal of Neurotrauma. So to test this, we expose slices to level 4 blast exposure or sham control conditions. Using Western blotting techniques, we quantified the effect of blast on expression of key proteins for the LTP induction pathway at 24 hours post-injury. We observed the effect of blast in cultures when LTP was not induced, and in cultures when LTP was chemically induced. To chemically induce LTP, we exposed cultures to glycine, which has been commonly used in literature as a surrogate for electrical LTP induction, as both induction methods similarly activate NMDA receptors. We observed that the time course of glycine LTP deficits following blast exposure matched the time course for electrical LTP deficits presented in aim 2, which adds greater confidence to the similarities of those induction methods. The proteins we targeted are all directly involved in LTP induction, which we will dive into right now. So the cellular process of LTP occurs at the synapse, where one neuron connects to the next neuron, specifically in dendritic spines, diagrams of which are shown here and in each of these images to the right. Alpha calcium calmodulin dependent protein kinase 2, or CAMK2 for short, senses the calcium influx triggered by activation of the NMDA receptor. You can see calcium influx here, and activation of the, or phosphorylation of the CAMK2, or CAM kinase 2. The kinase translocates to the postsynaptic density, which is this motion here, and binds to the NR2B subunit of the NMDA receptor. Previous studies have shown that the NR2B subunit was identified as the mechanosensitive NMDA subunit in stretch injury studies. Now that CAMK2 is at the synaptic membrane, it can phosphorylate our next target, the AMPA receptor. LTP is hallmarked by insertion of excitatory AMPA receptors into the area known as the postsynaptic density, which is highlighted here and in this faint yellow color in each of these images. Phosphorylation of the GluR1 subunit of the AMPA receptor increases AMPA receptor ion conductance, which was occurring here. You see this phosphorylation site. And it also increases extrasynaptic receptor priming, which refers to an increase in the number of available AMPA receptors outside the synapse that are primed for incorporation into the membrane, which is being shown here. Both of these are necessary steps for the induction of LTP. The next target is postsynaptic density protein 95, or PSD95, which is shown here in blue and here in this large yellow oval. This is the major postsynaptic density scaffolding protein, and it immobilizes AMPA receptors at the synaptic membrane so they can mediate the increase in signal that leads to LTP. PSD95 immobilizes AMPA receptors by binding to the protein known as stargazin, which is the final protein we'll be investigating, Stargazin is an auxiliary protein that is attached to the AMPA-GluR1 subunit and can bind to PSC95 when stargazin is phosphorylated, which you are seeing. This is stargazin attached here, and it's binding to PSC95 there. You can also see it occurring there. So now if we move to the Western data, we first observed that there was no effective injury on CAMK2 or NMDA NR2B subunits. This indicated that CAMK2 could be activated after injury, and that there was no lack of NR2B subunits for CAMK2 to bind to at the synaptic membrane, which suggested that the problem was linked to the number of AMPA receptors that are immobilized at the synapse. So in A, we're looking at the phosphorylation level of the GluR1 subunit at the serine 831 site normalized to its loading control beta-actin. Phosphorylation of this site increases AMPA channel conductance. However, AMPA receptors must be immobilized at the synapse for this phosphorylation event to occur. We are comparing the effect of blast on non-LTP and glycine LTP cultures. Here we see no effective injury on the non-LTP cultures. We also observed an expected increase in phosphorylation in sham cultures with LTP induction. However, when LTP is induced, phosphorylation in blast-injured cultures is reduced as compared to sham, shown here. In B, we report expression of total GluR1 normalized to beta-actin. Again, we see no change in non-LTP cultures. However, when LTP is induced, blast prevented an increase in total GluR1 expression as compared to sham. In C, we reported phosphorylation of GluR1 subunit at the serine 845 site, normalized to beta-actin. Phosphorylation of this site increases the extrasynaptic receptor priming, again referring to that pool of AMPA receptors that are available outside the synapse and are ready for incorporation upon LTP induction. Here we see no effect of blast on non-LTP cultures. With LTP, phosphorylation is increased for both sham and blast cultures. This indicates to us that the cellular machinery responsible for AMPA receptor priming was still functional. We also observed that blast reduced PSC95 expression and stargazin phosphorylation. In A, we're reporting total PSC95 expression normalized to beta-actin. Here, blast injury reduced PSC95 expression for both non-LTP and glycine LTP cultures. It's important to note that this is the only target that we investigated where it was reduced regardless of LTP induction. In B, we reported phosphorylation of stargazin at the serine 239-240 site, normalized to beta-actin. Here we see no effect of blast on non-LTP cultures. For glycine-induced LTP, we see that phosphorylation in the sham group increased as expected. However, blast injury prevented this increase in stargazin phosphorylation. So to summarize the same, we found that primary blasts influence multiple synaptic targets that are critical to LTP, including GluR1 and stargazin upon LTP induction, and PSC95 regardless of LTP induction. There was no effective injury on the serine 845 phosphorylation site on GluR1, CAM kinase 2, or NMDA NR2B receptors. Excuse me. The fact that PSC95 expression was reduced regardless of LTP induction suggests to us that PSC95 degradation was the driver of deficits downstream. And in this graphic to the right, we see depictions of an uninjured dendritic spine and a blast-injured dendritic spine. After blast, PSC95 degrades, which we have indicated as going from this darker pink color to this lighter pink color. When the cell receives signaling for LTP induction, primed AMPA receptors, shown here to the right, AMPA receptor stargazing complexes can still move through the synaptic membrane. However, stargazing and PSC95 cannot link, thus AMPA receptor immobilization at the synaptic membrane is prevented, represented as less light green triangles on the injured spine. CAM kinase 2 is present at the membrane and ready to phosphorylate GluR1 containing AMPA receptors. However, since they are still mobile, this phosphorylation event cannot occur, thus preventing an increase in signal that's normally witnessed with LTP. Now, non-blast TBI studies have observed that increasing the secondary messenger known as cyclic adenosine monophosphate observation